In today's video, we will explore a promising discovery in cancer research involving a sugar compound from deep-sea bacteria. This bacterium can cause cancer cells to self-destruct through a process called pyroptosis. It examines how this compound, EPS 3.9, works, how it compares to traditional treatments, and its potential future in immunotherapy. Just a friendly reminder to hit the like button if you enjoy this video. I would appreciate it very much, and it's free. Warning, not everyone will find this interesting, but let's begin! Recent scientific discoveries are revealing how substances found deep within our oceans might hold the key to novel cancer treatments. Among these findings is a breakthrough involving a sugar molecule produced by marine bacteria that has shown the ability to destroy cancer cells explosively. This sugar molecule, known as EPS 3.9, is a long-chain exopolysaccharide composed of the sugars mannose and glucose. It was found to induce a powerful form of cell death in cancer cells called pyroptosis. Pyroptosis differs from other types of cell death in that it is both inflammatory, destructive, and it alerts the immune system to the threat. Early laboratory tests and animal studies suggest that this sugar is a potentially powerful dual-action approach to cancer treatment. This promising research was publicly announced in July 2025 and published in the FACEB Journal, a respected scientific journal focused on experimental biology. The work was carried out by a team led by Dr. Qiumin Sun at the Chinese Academy of Sciences. The scientists focused on a strain of marine bacteria named Spongibacter nanhinus CSC 3.9 found in deep-sea environments. From this organism, they isolated the sugar compound EPS 3.9 and began testing its effects on cancer cells. The research is still in its preclinical stages, meaning no human testing has occurred yet. However, the effects have already been observed in two important models. In laboratory experiments using human leukemia cells and in mice with liver cancer, the compound triggered pyroptosis in the tumor cells. The result was not only a reduction in tumor growth, but also a marked activation of the immune system. This suggests that EPS 3.9 could potentially serve a dual purpose, directly eliminating cancer cells while also training the immune system to recognize and attack them more effectively. Conventional cancer treatments, such as chemotherapy and radiation, typically aim to stop cancer by damaging the DNA of cancer cells or halting their ability to divide and grow. While often effective, these treatments are known for their severe side effects, such as affecting healthy cells that divide rapidly, the digestive tract, hair follicles, and bone marrow. By inducing pyroptosis, it prompts cancer cells to undergo a highly targeted and inflammatory form of programmed cell death. This process not only reduces the risk of damage to nearby healthy tissues, but may also be more effective in attacking tumors that have developed resistance to standard treatments. Unlike many chemotherapy agents, EPS 3.9 doesn't merely poison the cells, it causes them to rupture from within, avoiding some of the escape mechanisms cancer cells use to survive treatment. Another important distinction is its interaction with the immune system. Many conventional therapies do not engage the immune system at all, or may even weaken it. In contrast, EPS 3.9's method of killing cancer cells activates immune cells and increases their ability to target tumors. It acts more like modern immunotherapies, which are designed to harness the body's natural defenses to fight cancer. This is just one example of an emerging class of potential cancer-fighting compounds derived from marine environments. Other ocean-based sugars, including those extracted from sea cucumbers, have demonstrated anti-cancer properties in laboratory studies. Some of these compounds work by blocking enzymes that cancer cells use to invade other tissues and spread throughout the body. These findings highlight the broader potential of marine organisms as sources of new therapeutic compounds. However, bringing these marine-based treatments to market presents significant challenges. Marine sugars are often complex and difficult to synthesize in large quantities. Scientists must figure out how to produce these molecules consistently and affordably. This involves developing specialized manufacturing processes or finding ways to reproduce them using biotechnology. These technical hurdles must be overcome before any marine sugar compound can become a commercially available treatment. One of the most exciting aspects of the research on EPS 3.9 is its potential to enhance immunotherapy. This is an area of cancer treatment that has gained momentum in recent years. Many tumors are considered cold, meaning they don't provoke a strong immune response, making them harder to treat with immune-based approaches. By triggering pyroptosis, it turns these cold tumors into hot tumors that actively signal the immune system to respond. This mechanism could allow it to be used alongside existing immunotherapies, potentially boosting their effectiveness. For example, combining it with checkpoint inhibitors or T-cell therapies might produce a stronger and longer-lasting anti-tumor response. The idea of using pyroptosis to amplify immune engagement is gaining support in the cancer research community. Compounds like EPS 3.9 of May become a valuable part of multi-pronged treatment strategies in the future. Although the early findings are promising, it is not yet ready for clinical use. At present, its effects have only been tested in laboratory settings and mice. The path from preclinical research to approved medical treatment is long and complex, requiring extensive safety testing, clinical trials, and regulatory approvals. The next steps in developing EPS 3.9 include conducting more comprehensive animal studies to assess toxicity, identify which types of cancer are most responsive, and establish safe dosing levels. Researchers must also figure out how to produce this in sufficient quantities for large-scale testing. If those early hurdles are cleared, the compound will enter a series of human clinical trials. This typically starts with small groups of patients to ensure safety before moving to larger trials designed to evaluate effectiveness. The entire process, from preclinical research to potential approval, can take anywhere from 5 to 10 years or more. Much depends on the early results of animal studies and initial human trials. Even if it proves to be safe and effective, manufacturing, distribution, and cost-related issues will also need to be addressed before it becomes widely available. The discovery of EPS 3.9, a sugar compound derived from deep-sea marine bacteria, offers a potential new path in cancer treatment. This triggers pyroptosis, a powerful, inflammatory type of cell death. First reported in July 2025 by scientists at the Chinese Academy of Sciences, this research is still in early preclinical stages. It has already shown effectiveness in lab tests and mouse models. Unlike traditional treatments, it not only kills cancer cells, but also activates the immune system. This makes it a valuable addition to immunotherapy-based strategies. Though still years away from human use, EPS 3.9 highlights the untapped potential of marine biology in the fight against cancer. This could one day lead to safer, more targeted therapy in future treatments. So, what do you think of this video? Leave me a comment and check out some of my other videos. Don't forget to hit the like button and subscribe. Thanks for watching, and we look forward to seeing you at the next one.